Well, it's a Spartan audience, so I'm open for questions and interruptions as we go. My name's Tom. I'm from the University of Utah. I'm in the Department of Medicinal Chemistry there. And we've had Blue Waters allocations for a number of years now, also large exceed allocations and others. And what we'd like to be able to do is model RNA structure, ideally. And this is complicated because RNA is inherently dynamic. And when you see motion in the simulations, you don't know if that motion is real or is an artifact of the force field without the ability to assess and validate these force fields and what they do. And what we found over the years, the force fields are not necessarily reliable. But we need fast, accurate simulation methods. We need validated force fields with a proper balance among the interactions between both the intramolecular and also their interactions with solvent and ions. We need good simulations, and that's difficult in the case of RNA because they're dynamics. And as I mentioned yesterday in the paid presentation, we have a strong focus on trying to assess convergence and reproducibility in these simulations. And as a way of introduction, any short simulation from an experimentally valid structure will stay near that experimental structure for a period of time and give you good reproducible results that represent the experiment. But invariably, if we run the simulations longer, we often find we move to a new place that perhaps may be lower in free energy, and you get trapped there and you can never come back. And this has been the history over the last 20 to 30 years in my work with MD simulation, that we keep finding these problems as we run longer. We modify the force fields to fix them. They work for a period of time, and then we break them again. Now, with the access to facilities like Blue Waters or David Shaw's Anton machine, where we can do on Anton, we can run very long simulations. On Blue Waters, we can run ensembles of independent simulations. And we can show that the ensembles of independent simulations show similar convergence properties with respect to regular long simulations. And we can get independent simulations with different initial conditions to give reproducible results and converge. And I've talked about the implications for DNA recognition in the past. I don't want to talk about it today. Just want to show some slides I showed last year on how well the force fields can reproduce DNA structure. And kind of the red and blue lines are average RMS deviations away from average NMR structures for the Dickerson dodecamer. And they represent the Barcelona 1 force field, BSC 1, and also the OL15 force field from the Czech group. And they're amazingly close to the experimental structure. So we look at these two force fields, Barcelona 1 or OL15, in different water models. For the OPC water model with the OL15 force field, the average structure that we get from our simulations is within 0.44 angstroms of the average NMR structures. It's remarkably close. It's really kind of surprising we can get so good. If you look at the overlap of these structures, blue and red again represent the two force fields that are probably the best ones in the amber suite at the moment, and black is the experiment. You can really see that deviations are not so profound in terms of how these base structures overlap. If we add in the Barcelona 0 force field, you can see it's starting to deviate away from those two other force fields. So those two force fields are really amazing. If we look at CHARM, deviation's much more severe in terms of the CHARM 36. Now, this work I haven't talked about in the Blue Waters before. We've been playing with these particular anti-cancer compounds from a Mexican group that synthesized a lot of these. My postdoc, Rodrigo, brought some of these compounds with him from Mexico. They're copper-containing compounds with various aromatic things. They bind better to various, as far as their activity against anti-cell lines, they're better than cisplatinum in some cases. And there's alternate theories on why these compounds are active. Maybe they generate reactive oxidative species, or perhaps they affect the DNA nuclease activity. So we wanted to use simulations to kind of see how these compounds would interact with DNA. First paper was out in 2015 that talked about this in more detail with 21 copper compounds. But digging deeper, the ability with Blue Waters to run a whole set of lots of simulations of the DNA with different initial conditions, multiple of these compounds sitting in the solution with the DNA, we could see events that I wouldn't have expected to see on the time scale of the simulations. These simulations are on the order of 20 microseconds each. We saw stacking of the compounds on the ends of the DNA. That makes sense, an aromatic pi-pi interaction. We saw minor groove interactions, which we expected, but we were surprised by, in 4 of the simulations out of 36, we saw spontaneously the drugs could move into the DNA and force the base pairs out, this base pair aversion mechanism. And this is kind of remarkable because internal base pair opening occurs on a millisecond timescale, not a microsecond timescale. But on the order of microseconds, we're able to, in the simulation, spontaneously see this. We could also see a spontaneous intercalation event on the end because the base pair is frayed, the drug came in and stacked, and the base pair is closed on top of it. So it's kind of amazing that we can start seeing some of these effects that go beyond what we would have previously been able to do. In the simulations where we see this base pair aversion, kind of a consistent mechanism, it takes on the order of 100, 200 nanoseconds for this thing to occur. And then we can now look at the influence of different ligand substitutions on these compounds to see how it affects their interactions with the DNA and therefore may affect the activity as anti-cancer. So 2 examples here with different substitutions, running with the OL15, lots of independent simulations. And we can kind of see where these compounds are binding. This one's kind of sticking in the minor groove mostly, not doing so much of the aversion. And we can characterize for the different systems what the drugs are doing and hopefully provide insight. And what's interesting, too, is, I said my postdoc, who's not really done experiments before, brought some of these compounds back. So we're actually now doing the experiments to assess these things, DNA footprint analysis, how it affects the endonuclease activity, NMR titration, some QM calculations as well. And I never thought my lab would kind of get involved in the experiments to assess these things, so it's kind of interesting. 4 minutes, 5 minutes?
No, I was saying.
Oh, question, yeah.
The compounds that he brought back, why are they something that we didn't know about here, or is it from a lab down there?
It's from a lab down there that synthesized them, so they're not a commercial. So this lab just has an expertise in developing these copper compounds and testing them. We just thought it'd be interesting to see how simulation would do with them. So what's really interesting to me is normally if you've got a metal-containing compound and you stick it in the NMR, it causes this paramagnetic line broadening and stuff, and you can't get a good NMR signal, particularly with copper. But we're able to get, these are one-dimensional NMR traces, that we're able to get shifts in the peaks without the line broadening. So we're now doing 2D NMR to kind of solve the structures of these things, which is kind of fun and neat. This is just hot off the press. This is a different project. We're working with Steve Harvey here, where they're looking at kind of DNA transport through pores. And we built a virtual cylinder. It's just a hydrophobic cylinder. And then we have bands of charges that expand and move up and then move back to see its influence on the DNA translocation and its properties. And there's theories about how the DNA may either extend or be scrunched by these charges in this band. Theories that were put forth via protein DNA transport mechanisms by Harvey, and so he wanted to see if we could reproduce some of their hypotheses in simulation. And we can see if we charge up these cylinders, like the positively charged similars cause the DNA to shrink because you got the negative charge of the phosphate, and do the translocation. So we're able to see this in simulations, and the charges, the +2, the +5, +10 are the magnitude of that charge change. So the larger the magnitude of that charge change, the greater effect it has, the more scrunching you see. And when we do negatively charged, you extend the DNA. So this is kind of neat, and again, one of these things that's made possible by having access to blue waters and be able to run these large ensembles of simulations. Okay, back to RNA. I talked a little bit about this last year. 2 different RNA systems. The names or details are not so important, but it's the Varkud satellite ribozyme stem loop 5 and also the GTPase activating center RNA. They're 2 particular RNA structures. We talked about this previously because it was published in 2015. We have a new paper as well, that we wanted to see if we could reproduce the magnesium-dependent conformational changes in this particular RNA structure. And there's known binding sites where these things bind. We showed that if we re-refined the structure with the original NMR data in explicit solvent in the modern force fields, we got a much better representation of the structure than previously. I've talked about this before. We were able to show that we could see spontaneous transitions in the structure due to magnesium, effectively an ordering of this loop structure that's shown there by the addition of magnesium. But I just want to talk about it again from the cautionary tale, that we could see successful transitions from the magnesium-free structure to the magnesium-bound structure sometimes. And that depends in part on the ion parameters. If one of the magnesiums binds to a particular site in the RNA and chelates with a phosphate group, you trap the structure. It's not going to be able to do the conformational change. And the problem is, with this new 1264 ion parameter from Kenny Mers's lab, which adds in a R to the minus fourth van der Waals type term to improve the performance, they get the correct binding affinity for a magnesium to the phosphate. That's about 12 to 13.5 kcals per mole. But if you just look at transition state theory, you want to break that bond, that's many milliseconds of time it's going to have happen. And we're running our simulations with a microsecond time scale. There's no way you could, if you're trapped, recover from that trap with standard MD methods. You'd have to go to some canonical, grand canonical, or do some tricks to kind of pull it off. Or maybe the force field shouldn't be accurate. Maybe we should make it so that it's easier to pull off than reality. And if we do that, like the Nielsen parameters, we can still see the correct conformational change, and we no longer get trapped. So we're kind of doing a cheat in the force field to allow us to get the magnesium properties. Now we've moved on from that and looked at the kissing loop junction of this stem loop 5 with stem loop 1 in simulations as well. It's nice and stable in the simulations, doesn't really fall apart. For magnesium-bound structure, we get a reasonable agreement between calculated residual dipolar couplings to experimental residual dipolar couplings. The different colors are different simulations. We've done lots of different independent simulations with different initial conditions. We wanted to look at the effect of magnesium, and if you pull the magnesium away, the structure is more flexible, as indicated by a larger dispersion in the difference between experimental and calculated NOEs. But what's interesting to us, although the magnesium orders the structure a little bit more, there's no real difference in the stability of the structure as estimated from an MMGBSA type approach. Whether we have no magnesium, we have magnesium, or we have magnesium bound where it's supposed to be bound with excess magnesium, there's effectively no difference, which is kind of a surprise. What we do see, though, what's interesting to us is that the stem loop 5 is very, very rigid, whereas the stem loop 1 is quite dynamic. It's a little less dynamic with magnesium present, but it adopts multiple different conformations. And what we think is the magnesium promotes the stem loop 5's role as a structure for kinetic capture of the more mobile stem loop 1, and this agrees with experiments that show that this kissing loop junction precedes the folding. The junction formation precedes the loop folding of the stem loop 1. Okay, in a related system, this GTPase activating center RNA, this one, on the other hand, is very sensitive to salt. It's got a bound potassium and some bound magnesiums that are seen in the crystal structure. Some controversy about whether it's potassium there or sodium or magnesium at that potassium binding site. But if you just run standard simulations without salt, the RNA blows up very quickly. That's what we would like it to do. That's what we'd expect. If you look at a PMF of pulling away these two loops, basically the energy goes down when there's no ions there. And if we add in neutralizing ions with an exclusion area around that particular important binding site between the two loops of the ions, you see as we get closer and allow ions closer and closer to that particular binding site, we suddenly get to the point where we're now stabilizing the bound stable structure versus the unfolded structure. And so we can kind of get these kind of estimates out. And We also show that without the chelating magnesium in its particular binding site, effectively your isoenergetic folded versus unfolded, that's on the right. But when we include the chelated magnesium and the potassium, we now clearly stabilize the folded state over the unfolded state. Okay, so I haven't talked a lot about the problems we have with these methods this year, but there is a clear force field misbalance. If we run simulations long enough of tetraloops or of other things, we will find alternative structures that are not representative of experiment. And this has to do with subtle balance among the intermolecular interactions, the interactions with water, the interactions with iron. And it's a difficult problem to fix. We've been working on it for many years. Sampling, we can get trapped in particular conformations. I'll show a quick example of that before I run out of time. I talked about this last year a little bit, that force fields, methods, interoperability, there's some issue that when you run the amber force field in GROMACS, it may not be the same as running the amber force field in AMBER or the amber force field in NAMD due to subtleties in the implementations that could have influence on the results. And judging conversions or overlap of simulations from different groups is tricky, and it's something we should be doing more often, is that we cross-correlate our results with another simulations group to show that we're all on the same page. But here's an example of trap conformations. Temperature replica exchange wasn't converged, and what happened is if we look at the RMSD histograms for each individual replica, they should be identical among the independent simulations, but they're not. There's this peak at 4 angstroms that occurs in 2 to 3 of the replicas, depending on the simulations. And what happened was, despite going from low temperature to high temperature in the replica exchange, we were just trapped in a particular conformation. This particular one, that's a funky one. As I mentioned last year, we can now converge results for different systems like a tetranucleotide with all the different force fields. There should be peaks at about 2 and 3 angstroms that are dominant structures, but we see all the force fields show these anomalous peaks further to the right that are not representative of reality. I mentioned in the PAID that to process these results, we had to really modify our analysis codes, so we wouldn't take out the luster to make it so we were doing full parallelization across the file system to process these ensembles. And we've got this 4 levels of parallelism built into the code. Other nice features, we can converge the tetraloops as well with different force fields, but none of them are correct, unfortunately. And even in multidimensional replica, it takes a long time to converge the distributions, probably on the order of, depending on the system, anywhere from, for tetranucleotide, it takes about 200 nanoseconds per replica for 192 replicas. For tetraloop, 360 replicas, it's about 2 microseconds per replica to converge it. So that's a lot of compute time to converge these things. I'll just mention briefly 1 minute on the Ebola project. Where we are with that was kind of designing peptides that from known structures that prevent viral entry, kind of developing the software framework to build up these potential structures using Rosetta, equilibrate them, minimize them, run MD, and then do free energy analysis. And we're getting that now all automated to run within the 48-hour queue limit in Blue Waters. And one real problem was when the student tried the first time, he was overloaded with data. He couldn't figure anything out, and then the data got deleted from scratch, and I yelled at him about that. So we're rerunning those now, and what we've done is we've developed a database framework with SQLite that essentially, from each simulation as it's running, pre-populates analysis on that simulation and sequence. So, and that puts it in a file in Blue Water, so we can just transfer home, and then we can go search on that and do that. And that now is automated, this whole data handling. So we don't even really save the trajectories anymore because we could just rerun them if we want to do that. So I'll stop there. It's kind of a quick, eclectic presentation of different things. But thank the computer time on Blue Waters, Exceed, and locally, and even on Anton from a few years ago. And these various people up there that many of whom have moved on now. So I'll answer any questions.